In the first class of human reproduction, we had already learned about the male reproductive system and the female reproductive system. Now let us move forward to know how gametes are formed. Now, as we all know, during sexual reproduction, there is union of gametes, which we name as a male gamete produced by the male individuals and a female gamete produced by the female individual. The process of gamete formation is known as gametogenesis. Now, the events which lead to formation of gametes in each of the individuals, that is male and female, differs. In humans, here we are talking about it differs. So let us try to know how this gametogenesis actually occurs. So gametogenesis, that is formation of gametes. Now in females, the process of gametogenesis is known as Oogenesis. That is the formation of the female gamete, which we also call as ovum or egg, is also, or even it is known as oocyte, is known as oogenesis. And in males, the same process, that is formation of male gametes or the sperms. Okay, male gametes or the sperms or spermatozoa is known as spermatogenesis. Genesis means formation, origin. Okay, so oogenesis and spermatogenesis. In females, where does it occur, or where does the formation of gamete take place? Female gamete always forms inside the ovaries, the two ovaries which are located in the lower abdomen, as we saw in our previous lecture. So it is formed in the ovaries, and in males it is formed inside the seminiferous tubules inside the testes. In each of the testes there are testicular lobules, and inside the testicular lobules are present that long tube, highly coiled tubular structures known as seminiferous tubules, and inside those tubules there is formation of the male gamete or the sperms. In females, it is a cyclic process which is repeated at an average interval of 28 days. In human females, okay, the cycle that is followed is known as menstrual cycle. Okay, it is known as menstrual cycle. So it repeats. It is repeated. That means after every 28 days, there is formation of one egg and its release. Again, that cycle gets repeated after 28 days. Now here 28 days is an average value that is taken. It's an average value. Always there will be plus or minus number of days. And in males, it is not a cyclic, but it is a continuous process. That means continuously the seminiferous tubules will be forming the male gametes or the sperms. It is not a cyclic. It is not repeated in a cyclic manner, but it is a continuous process since the onset of puberty till the time the male individual remains capable of producing the male gametes. Then in females, one very interesting fact in females, human females, is that the oogenesis, the start of oogenesis takes place when that particular female individual, she herself was a fetus. She herself was a fetus inside her mother's womb. Okay, so it initiates in the fetal stage itself, but the initiation of oogenesis, that means the formation of the gamete, takes place there itself, when she was just 25 weeks old fetus inside her mother's womb. But the egg, the release of the female gamete or the egg or the ovum, it starts only when she attains puberty. And that attainment of puberty, the first release of egg, is known as menarche. But in males, it initiates at puberty. Unlike females, it initiates at puberty. That means the division process itself starts at puberty. Then in females, the ovum release, it stops Okay, after a certain age, it stops. The ovum release stops. It starts at puberty when we call it menarche, and it stops after a certain number of years, usually 40 to 50 years. But even in this case, they can be sometimes beyond 50 also. It may continue the full release of egg, and later after 50 plus, it may stop. So, or less than 40 also, it may stop the release of ovum. So, whenever it stops, we call it as menopause. The formation of female gamete stops. So after that, that particular female will no more be able to reproduce. Now in males, unlike females, in males, it continues uninterrupted until death. The formation of gamete, it continues until death, unless and until the particular person is suffering from some ailment or disease. That is a different issue. It might happen so, but otherwise normally it will continue. That means the males, they remain capable of reproducing or releasing sperms, okay, till death. Then there is release of only one ovum at a time in females. In males, release of millions of sperms take place at a time. Okay, release of millions of sperms takes place at a time. Whereas in case of females, it is only one ovum at a time. In a period of 28 days, one ovum will be released. Now we will go into the process of spermatogenesis. Now spermatogenesis, as already mentioned, it is the process of formation of the male gametes which initiates at puberty. So here we have the figure. Okay, here we have the figure which highlights the formation. Now here, what is this? This is the testis. This is the figure of the testis which we already talked about yesterday. These are the testicular lobules. Okay, these are the testicular lobules. Okay, so here is a testicle, and each testicular lobule, what does it contain? It has the seminiferous tubule. So you can see here, this is one seminiferous tubule which is enlarged, the internal structure of a seminiferous tubule. Now, these things were also talked about yesterday. Each seminiferous tubule, they have two types of cells, that is Sertoli cell and the spermatogonia. Now spermatogonia are the cells which are diploid, which has 46 number of chromosomes, but they undergo meiosis. They are capable of undergoing meiosis or reduction division and form the sperms. Okay, so here from the inner side of the membrane of seminiferous tubule, okay, the spermatogonia start emerging, and as they divide, they move towards the center, so that finally when they are fully developed into sperms, they will remain in the central portion. So a small portion enlarged here, as you can see here. Okay, it's enlarged. These are the spermatogonia which are diploid. Okay, then they will undergo mitotic divisions. Okay, so here spermatogonia which has 46 chromosomes, they will undergo mitosis. Okay, first mitotic division, each of them will undergo just to increase the number. Okay, and those new cells will be called as primary spermatocytes. Still here, the chromosome number is 46. Then after that, the primary spermatocytes, they undergo meiosis. Okay, they start with meiosis. So if it is asked to you which of the cell undergoes meiosis or starts with meiosis, then your answer should always be primary spermatocyte. So it undergoes meiosis. Now I think all of you remember that meiosis is divided into two phases, meiosis 1 and meiosis 2. Okay, it is divided into two phases, meiosis 1 and meiosis 2. So first it undergoes meiosis 1, the primary spermatocyte, and it forms new daughter cells called as secondary spermatocytes. And I hope you can recall, meiosis 1 results into daughter cells which have half the number of chromosomes. To each. One primary spermatocyte will result into two secondary spermatocytes. As you can see here, two secondary spermatocytes. Now each of them will have half the number of chromosomes, that is only 23 and 23. Now this secondary spermatocyte will undergo the second meiotic division to form the spermatids. They will form the spermatids. Now it will continue with the half the number of chromosomes, just the number will increase here. That is second meiotic division. That will result into spermatids. How many? 4 from each primary spermatocyte. One primary spermatocyte forms 2 secondary spermatocytes, and then each of the secondary spermatocyte will form 2 spermatids. So total it will be 4. As you can see here, these are 4 from one primary spermatocyte. Each of them will be having the haploid, that is 23 number of chromosomes. Now each of these spermatids will now modify, okay, will mature, will modify its structure to form the sperms or spermatozoa. These are the male gametes. These will behave as the male gamete. And the process of the development of spermatids into spermatozoa is known as spermiogenesis. Okay, this is known as spermiogenesis. Till spermatids, it is the division phase, and after the spermatid formation, it is the developmental phase, which is known as spermiogenesis. Develops into the male sperms or the male gametes, also known as spermatozoa. So this is about spermatogenesis. Spermatogonia, developing to form primary spermatocyte, which divides to form secondary spermatocyte, spermatids, and finally the sperms. So here again we have the same thing, it's repeated. So this is the testes, these are the testicular lobules, epididymis, retrotestis, vas efferens, vas deferens, everything is located here. Now once the seminiferous tubule, so this is the seminiferous tubule, this is how they, the spermatogonia, how they have divided. Okay, this is primary spermatocyte, secondary spermatocyte, spermatids, and spermatozoa. So as they develop, they move towards the center. And the Sertoli cells, which are located in between the spermatogonia, help in all this development by providing food, by providing nourishment. So the structure of one sperm cell. Okay, this is the structure of one sperm cell. This is how they modify. So here you have one more figure we have here of spermatozoa or sperm. Now it had developed from spermatid. Spermatid was just a normal animal cell. Now how did they modify structure? What happened? They have a bulbous structure which is known as the head. It is called as head. Now head will contain the nucleus, which will have the 23 chromosomes. The tip of the head is known as acrosome. This tip has a lot of significance during fertilization. It contains digestive enzymes, also known as sperm lysins, because this is the structure which, when comes in contact with the egg, will dissolve the membrane of egg and let the chromosomes from the sperm enter into the egg to form the zygote. So acrosome has a lot of significance. Then followed by the head portion is a slender structure which is known as the middle piece, which will contain many mitochondria. Why many mitochondria? Because a lot of energy is required by the sperm. Why a lot of energy is required by the sperm? Because after the release of sperm, it has to travel. Okay, it has to travel from the male reproductive system to be deposited into the female reproductive system, and from the female external genitalia, it has to move through the cervical canal, through the uterus, through the fallopian tube to finally meet with the egg in the ampulla isthmus junction of the oviduct. So that journey, that entire journey, okay, till the time it can fertilize, that journey has to be covered by this tiny sperm. So it needs a lot of energy, and that energy is provided by these numerous mitochondria located in it. Then the plasma membrane extends as a long tail-like structure, flagella. Okay, it is called as flagellum or flagella, long tail-like structure called as tail itself. Okay, this is just the extension of plasma membrane, which will help in motility of the sperm, which will help in movement of the sperm. So this is the structure of spermatozoa or sperm. This is another figure. The figures like slightly diagrammatic views we can vary. So both of these represent the same, that is structure of sperm. Now hormonal control of male reproductive system. That is, how does the male reproductive system function? Now hypothalamus, which is located in our brain, okay, which is located in our brain, releases gonadotropin releasing hormone. Okay, gonadotropin releasing hormone, that is GnRH, which will stimulate the pituitary gland, to release luteinizing hormone and follicle stimulating hormone. These two will be released by the pituitary gland, the anterior part of the pituitary gland. The LH that is released by the pituitary gland located in the brain will move through the blood. These are endocrine glands, will move through the circulatory system and finally reach upon the male reproductive system in the Leydig cells. Leydig cells, where are they located? They are the interstitial cells located in between the seminiferous tubules. So they will stimulate, this LH will stimulate the Leydig cells to release testosterone or androgen, the male hormone. This male hormone will then further stimulate the reproductive organs, tract and other organs. They will be responsible for all the reproductive activities going on inside the male reproductive system. Like say, even the development of the spermatids, if the initiation of gametogenesis and everything will be related to the release of the hormones by the Leydig cells, and even other secondary sexual characters will also be related to the release of these hormones. Follicle stimulating hormone, what will it do? It will go and stimulate the Sertoli cells. It will again stimulate the Sertoli cells. Okay, these are inside the testes. This is happening inside the testes. So Sertoli cells will become active. Okay, that will stimulate spermatogenesis. Even this. Okay, Leydig cell will also stimulate spermatogenesis. They will provide nutrition. Simultaneously, Sertoli cells will also produce a hormone called as inhibin. Because this process, though we are saying it continuous, but of course it has to take place off and on. So Sertoli cells, what will happen? It will release hormone inhibin. Inhibin will in turn go and inhibit the formation of FSH, so that it will stop for some time, till the time the sperms are released. So this FSH secretion will be stopped. Accordingly, the hypothalamus will also be stimulated to inhibit the release of GnRH. So thereby what will happen? Again, furthermore, as the level of hormone inhibin will fall. Okay, well, why will it fall? Because here we are stopping the release of FSH. So the release of hormone inhibin will fall. As it drops, what will happen? There will be further release of FSH. FSH will stimulate Sertoli, will stimulate inhibin. Again, rise of inhibin, it will stop. So what happens? Its rise will stop the spermatogenesis will reduce. That means the rate of spermatogenesis will decrease, and again as it falls, it will increase. So this is how it is controlled, the hormonal control of male sexual functions. Hypothalamus releasing GnRH stimulates pituitary gland to release LH and FSH, which will further stimulate Leydig cell and Sertoli cells respectively, to further stimulate the spermatogenesis by the hormone testosterone. So this is the hormonal control of male reproductive system. Now oogenesis, when taking place in females. So here this is the structure of the ovary. Now as already I had mentioned that when a female, she herself was a fetus, okay, inside her mother's womb, then there is initiation of oogenesis. That is, all the eggs that she will release during her lifetime from her ovaries, they are initiated, okay, when she herself was a fetus. All the number of millions of eggs will start developing. Okay, millions of millions of eggs, millions of cells, they will start with the division of meiosis. They will start. Okay, that. So here in the fetal stage, meiosis 1 starts. So here you can see initiated during embryonic stage, which are called as oogonia. Okay, so numerous oogonia cells will be formed inside the ovary. Each of the oogonia, they will start with meiosis 1. Okay, they will start with meiosis 1 when we call them as primary oocyte. The primary oocyte have started with meiosis 1, but they do not complete. This primary oocyte has not completed meiosis 1. They have started with prophase. As you can remember, meiosis 1 is divided into phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. So they start with prophase 1 and it gets suspended. Okay, it gets suspended. It does not proceed anymore. So all the cells, they still have 46 chromosomes. Okay, but the initiation of meiosis has taken place and everything remains suspended. The fetus, they grow. Okay, they grow. The baby is born. The female baby is born. Okay, and then she grows and she attains puberty. When she's about to attain puberty, juvenile is that phase before sexual maturity, before puberty. As she grows, okay, as she grows, what will happen? As she is growing, this primary oocyte, they start forming some follicular layers. Here you can see primordial follicle. That means some coverings. Okay, some cells start growing around them. This primary follicle. Okay, it starts growing. Still in the, having 46 chromosomes. Secondary follicles start developing. Okay, here secondary follicles also start developing. Then that is still the meiosis has not resumed, which was suspended at prophase 1. Still it has not resumed, gradually going towards to restart with the process. Then what will happen? Meiosis 1 completes. So once secondary follicles have fully developed, so by that time meiosis 1 will complete. Now here you have to remember one thing: not all the primary oocytes that is present inside the ovary will be undergoing these changes. One at a time or few at a time, one by one. If few cells are in the primary follicular state, the rest are still in primary oocyte state. If few of them are in the secondary follicular state, the rest of them are still in the primary follicular state. So batch by batch it goes on happening. So with the completion of meiosis 1, there will be formation of tertiary follicle. You can see here they get covered by cells. Okay, they not cells exactly. This is one single cell. Okay, they get covered by layers. Okay, that is known as tertiary follicle, finally maturing to form the GRAAFIAN follicle. So once the GRAAFIAN follicle has formed fully, okay, then that will move towards the release of the nucleus here. Okay, release of the nucleus. The nucleus is what? Nucleus has 23 chromosomes. Okay, and we call it as the secondary oocyte. We will call it now as the secondary oocyte, which has already completed meiosis 1. Okay, only meiosis 1. The secondary oocyte, it is ready to be released out. Okay, and when it is released out, we will say the first time whenever it is released out, we will say it's attainment of puberty. So the secondary oocyte, okay, secondary oocyte inside the GRAAFIAN follicle. This is the GRAAFIAN follicle. Okay, there's a secondary oocyte. Now here, meiosis 1 will also always result into formation of two cells. Okay, it results into formation of two cells. One we call as secondary oocyte, other is called as the first polar body. Now, what happens? The maximum of the cell content will be deposited in the secondary oocyte with minimal content in the first polar body. So what happens? Gradually the first polar body, it simply dies out, leaving behind only one cell, that is the secondary oocyte, which matures and also is released out. Okay, released out from the Graafian follicle. It's released out from the Graafian follicle to the ovulated ovum. This is called as ovulation. The ovum is released. Where will it be released? It will be released into the oviduct. The fimbriae will be there to capture it. Will be there to capture it and then transport it into the oviduct. Now the outermost layer, okay, outermost layer, it is known as the corona radiata. The layer which is covering the secondary oocyte. It is known as corona radiata. Of course, now again here, layer by layer it is formed. Now the secondary oocyte, it moves to one side of the cell, leaving an empty space here, which is a fluid filled space, which is known as antrum. Okay, this is known as antrum, and the oocyte remains here covered by a layer known as zona pellucida. A zona pellucida layer is formed around the secondary oocyte. So when the secondary oocyte is released, it is released along with the zona pellucida and a few layers of this corona radiata. What remains back? The follicular layer. The follicular layer, the antrum, the fluid, everything remains back. This, whatever remains back, gets converted to a yellowish structure which we call as corpus luteum. It will start releasing progesterone. Okay, it will start releasing progesterone after that. So this is the basic sequence of oogenesis. So here, furthermore, same thing, it's repeated here. Let us see. Oogonium, okay, which divides, that is in the fetal stage. This is before birth, as you can see here. Okay, primary oocyte arrested in prophase 1. Started with meiosis 1, but got arrested here, got suspended, still with 46 chromosome. Then the baby is born, childhood, ovary is inactive. Then gradually, as she moves towards puberty, what will happen? The primary oocyte will start becoming active. One after another. You can see in this figure, if one has released, the other is still in the Graafian follicle stage. The previous one is still with the developing follicles, and these are the immature ones with the primary follicles. So they will develop one after another. Then the primary oocyte completes meiosis 1 to form the first polar body, which dies, and the secondary oocyte will then be released. Ovulation here you can see. Okay, it gets released here, and then the secondary oocyte had started with meiosis 2 but again got arrested. This also we have to remember, because meiosis 1 has to be followed by meiosis 2. So the secondary oocyte started with meiosis 2 but again got arrested here. Okay, it did not complete. In that state itself, it will be released out from the follicles. Ovulation will take place, and if fertilization occurs, then it will complete meiosis 2. If no fertilization, then it will never complete meiosis 2. Okay, so that part of course we will talk about later, but for the time being, so secondary oocyte, let me repeat, primordial follicle, that is before birth. Then after birth, the primary follicle will develop. Then furthermore, secondary follicle, tertiary follicle, they start developing mature follicle, when we call as the Graafian follicle. You can see here, this is the Graafian follicle. They rupture out, releasing the egg. Releasing the egg where? Into the oviduct. So here, Graafian follicle and ovum. Okay, let us see the structure of this. Is a microscopic view. This is the secondary oocyte, the developing oocyte, okay, which has the zona pellucida layer. Okay, a zona pellucida layer is developed around it. So here, this you can see here also. This is the nucleus with 23 chromosomes. Okay, then they have a layer which is called a zona pellucida layer. The zona pellucida layer also plays a great role during fertilization. It has a lot of significance during fertilization. Outer to the zona pellucida are cells of corona radiata, okay, which is meant mainly for protecting the ovum, and it's filled with cytoplasm. So you can see here, now this is inside the GRAAFIAN. This is only the ovum, and here it is inside the GRAAFIAN follicle. So secondary oocyte, this structure will remain towards one side to be attached to an empty space filled with fluid that is known as antrum. Then outer to the covering, the follicles, that is theca interna. Okay, we call them as theca interna, the layers of the GRAAFIAN follicle. So whenever it is released out, this oocyte will be released. It will be released along with the zona pellucida, okay, and the corona radiata. It will be released out, leaving behind this theca interna, which will then be called as corpus luteum. Now, hormonal control of female reproductive system. As like in males, even here hypothalamus releases GnRH, which will stimulate pituitary gland, okay, to release FSH and LH, follicle stimulating hormone and luteinizing hormone, which will work upon the ovary to release estrogen and progesterone. Estrogen release takes place from the follicles. The developing follicles will release estrogen due to the action of follicle stimulating hormone. Okay, so they will stimulate the follicles to release estrogen. More release of estrogen, the development of the secondary oocyte takes place. Once the secondary oocyte is released, then what will happen? The corpus luteum will start releasing progesterone. So progesterone release will further help in during the fertilization process and also development of the fetus. So they will finally work upon the uterus. The progesterone will work upon the uterus, show its action in the uterus. So this is the hormonal control of female reproductive system. So today we learned about the gametogenesis in males and females. Next, we will move on to fertilization process.